Oh, there you go. So, welcome to this Monday evening. My name is Dr. Sveta Silverman. I'm sure you know me. It's a Facebook Live and stuff. Today, I'm actually quite nervous because I'm doing a little literature search update on NRF2, on mitochondrial health, on NAD. And, all this literature search updates, they're very sort of, nerve-wracking as to how can I incorporate it and tie it up together. So every time I read about oxidative stress, now the interesting thing is what I'm showing you today is the header is the title of the paper that I have read and I have reviewed, and the all the papers that I've read are from October 2019, other than one paper that was published in 2018. So these are very, very fresh off the press science validated papers. So interesting is every time I read about oxidative stress, I find something new in the definition and the explanation. So oxidative stress, we know, is a result of imbalance of oxidative species of free radicals and the rate of their removal. So the thing is, we do need certain degree of free radicals because it sustains our cell life, our cell viability. Because if we have a bad cell, it has to be wiped out, and that is recognized via these free radicals or that degree of oxidative free radicals. So now, and this is normal. And we also, that's what I teach. So the first part of this slide to me is very important because it's about lifestyle. It's about us eating alpha tocopherol. What is alpha tocopherol? It's vitamin A. So eat your vitamin A, eat your beautiful oils, eat your olive oil, drink your olive oil, eat your olives, avocados, nuts and seeds. Well, ascorbic acid, this is our vitamin C. So we need the combination of what we eat, and we need the combination of what we make as glutathione or superoxide dismutase and other substances. Now, look, under pathological conditions, free radical levels overwhelm cellular antioxidant levels. And that's what creates damage. And it damages the proteins, the fats, the DNA that can lead to cellular toxicity, cytotoxicity, cell toxicity, genotoxicity, genes toxicity, and ultimately to carcinogenesis. Now, during physiological conditions, the organism relies on the free radical scavenging systems. Good. However, once the free radicals are overproduced or the clearance is weakened, it leads to the cell damage. Well, if it leads to cellular death, it's not a bad idea. We don't need bad cells. But if it leads to cell damage and not recognition of it and cellular replication, then it is bad. So now I came across a very, very interesting paper about a brain hemorrhage, acute intracerebral brain hemorrhage. But we know that after the brain hemorrhage, there is another sequelae that is called secondary brain injury. So that's when we need to start taking care of. Now, so there are several cellular sources of free radicals. Where do we accumulate free radicals in the cells? Via the organelles, so for example, mitochondria, or the cytoplasm, or different other components of the cells, or their misregulations, for example, lysosomes, which are our carburetors that may not be working properly. Let's talk a little bit about why aren't we able internally to clean that free radical damage or something. Because we also age. And then, see, now I'm trying to tie up mitochondria and NRF2, mechanism of aging associated with oxidative stress and mitochondrial dysfunction. Now, look, from the paper, beautiful paper on Parkinson's disease, aging is one of the significant risk factors for Parkinson's disease and correlate with oxidative stress, later indicating later role in the pathophysiology or pathology of Parkinson's disease. Well, the same thing, NRF2, that our cellular protective complex or our cellular guardian. Look at this thing, which is called NRF2-ARE. So what happens is, when we are bombarded with free radicals, there is like oxidative free radical barrage or electrophilic cellular barrage. It turns on that cascade via that cellular guardian NRF2 complex. So what happens is NRF2 activates another element, which is called antioxidant response element. And those antioxidant response element system upregulates what is called survivor genes. That's when the nucleus and the genes start making all this defense, activating that defense apparatus. So now that NRF2-ARE system is affected by aging, and it is affected in neurodegenerative diseases like Parkinson's disease, like Alzheimer's disease. Look at this. We see that 54 affected genes and 31 are downregulated through not enough of NRF2, not enough activation of this antioxidant response element. But now look at this. If there is not enough NRF2, then it embarks on our famous NAD. And then see, it regulates the expression of another defense apparatus through NAD. And then it embarks on the production of energy, as you see here. We don't need to get into details, or the citric acid cycle that literally gives us energy. Or on another cycle that also pentose phosphate pathway, you don't need to worry about it, but that gives us energy. So the NRF2 is not working, or we're overburdened. Then the proper genes are not upregulated. The NAD downregulated embarks on that deficiency, and it's kind of that of us small but vicious circle. Mitochondria. Mitochondria is known to regulate calcium level. However, well, guess what? What ties up with the calcium metabolism? Our probiotics, our microgut is so critically important in calcium metabolism because it promotes the calcium absorption. So we eat this calcium. I hope we eat the proper calcium, bioavailable and bioabsorbable calcium. And the probiotics are going to promote that absorption. So the mitochondria can utilize that calcium in all those cellular processes. And for example, calcium is so critically important in cardiac muscle, amongst other organs. Now, From NRF2, let's look into NRF1, or the mitochondrial booster, or that transcription factor, or that protein complex that turns on mitochondrial DNA. Well, guess what? I just told you about antioxidant response element genes that turn on those survivor genes. Well, NRF1 is actually intertwined with some of those genes. So look, so then there is, again, the interplay between NRF2 and NRF1 through this guy. So that's why we need everything, biogenesis by NRF2 in response to stress. Interesting. Now let's try to tie up NRF1 and NAD. NRF1, as you see here, in combination with the other guy, are actually, as we know, turn on the production of new mitochondria. Well, guess what is turning on and upregulating this guy? The NAD complex. So we need the NAD, we need the NRF1, we need the NRF2, we need the mitochondria. And then here's the confirmation. That is the quote. This is, I could not put this. Just per se that Dr. Albina Dinkova-Kostova, I call her godmother of NRF2, she became a mentor in years. I met her many times, as well as Dr. Thomas Kanzler, who is one of the world-renowned NRF tours, and he is researching sulforaphane, and thanks to Dr. Kanzler, he conducted or conducts human trials on sulforaphane or broccoli sprouts. But we know that broccoli sprouts that contain sulforaphane, that is an NRF2 activation. But what I'm trying to show you is this. So it's another NRF2, but look, consumption of broccoli-rich diet in association with prevention of prostate cancer, or taking care of inflammation and carcinogenesis. I mean, what I'm telling you, this is I'm basically reviewing the papers. It's not about treating, curing, mitigating diseases. It's something that makes biological sense. Now, let's keep this. Now, why did I put this slide? I put this slide for a reason. When I started talking to you about intracerebral hemorrhage and then the sequelae of that intracerebral hemorrhage with a secondary brain injury, when we talk about the brain, we usually talk, oh, brain cells, neurons and stuff. However, that is nearly not enough. Brain cells, our thinking cells, have to be supported by that brain microenvironment, or in our world, medical word is called microglia, and we know it by fancy words as oligodendrocytes or astrocytes. But the thing is, we also know in my world, there are two papers about oligodendrocytes and NRF2 activator, pretandem, but the role of this, what is called gliostroma, is in association with neurodegenerative disorders. And as you see here, for example, Parkinson's disease. Astrocytes. Astrocytes have a well-tuned antioxidant response system, those ARE, regulated by NRF2. And it has to look, the interesting thing is, which is more efficient than in neurons in brain cells. Like really? And this is our famous, the master antioxidant of the cells, glutathione. The significance of this master antioxidant in this brain or neuron supportive cells is so critically important. Astrocytes actually provide glutathione to neurons. How important is that? Critically important. Now, so inflammation after intracerebral bleed or brain bleed, see, that's what happens. It creates that absolute barrage of free radicals. But then it exhausts the ability of cleaning those free radicals. So then what happens is, then there is a damage to the cellular membrane through those fats. So then we need to take care of the cellular membranes and introduce our fatty acids back to me in the shape of fish oils, essential fatty acids. Because if we don't look, it can lead to brain injury. Now, this is interesting. I put this paper for a reason. Edaravone is a relatively new FDA NRF2 drug for ALS. However, now they're introducing edaravone, the NRF2 activator, in the intracerebral hemorrhagic brain bleed. Now, and you see that they say that it improves the post-cerebral hemorrhage score, the mental score. They actually don't know per se the activity or something, but edaravone has multiple side effects. As you know, I'm always preaching natural stuff. But it's just the validation where we go with an NRF2 activation. The proper NRF2 activation, the proper mitochondrial activation, the proper maintenance of cell membrane is so critically important. So now, again, if I'm talking about mitochondria or I'm talking about DNA, we have to talk about NAD. Again, that paper on cerebral hemorrhage, so look, nicotinamide mononucleotide is a precursor of NAD. It significantly reduced brain swelling, brain cell death, oxidative stress, neuroinflammation, and on and on and on and on. But look at the interplay. By promoting the activation of NRF2 signaling pathway and inhibiting oxidative stress. And that is the whole point of today's talk. I can't even call it a lecture. It's a talk. Like, we need to combine this because it all makes cellular sense. Now, Parkinson's disease. FDP stands for familial Parkinson's disease. And there is a sporadic Parkinson's disease. But what's interesting about Parkinson's disease, look, to date, mutations in over 20 genes have been identified to cause Parkinson's disease. And those genes are defective proteins, oxidative stress, mitochondrial dysfunction, and abnormal calcium handling. And I just covered why we need to turn on NRF2, why we need to turn off mitochondrial boost NRF1, why we need to do NAD, again, to improve mitochondrial function. And the good mitochondria is going to lead to proper calcium metabolism, calcium handling. NAD. NAD and aging. NAD is a major player contributing to aging and age-related diseases, and here what it is. You can read this, but look at this slide. Not enough NAD, and that what is going to happen. That is why we need to upregulate that NAD. Now, on a cellular level, look what is going to happen. Our famous age or longevity, our famous telomeres. The epigenetic alterations, mitochondrial functioning, inability to make the proteins, stem cell exhaustion, altered intercellular communication, or that is our brain activity, cellular senescence, and genomic instability. That is why we so need that NAD. And this is another slide. NAD precursors need more NAD, so it does not cause DNA damage, it does not cause senescence. But again, look, NAD is tied up with the previous concept that I keep bringing again and again and again of inflammation, aging, and oxidative stress. Now, NAD is tied up with a group of another proteins or enzymes which are called sirtuins. I'm just throwing this slide again to show that progression or that important circle, that important feedback, that loop of N is a precursor of NAD. It's one of these niacins of vitamin B3s. So if we have this, if we eat this niacin, or we may need to supplement this niacin, or we actually make this niacin, and we recycle that niacin, so then it's going to lead to upregulation of, I'm showing you one of the sirtuins out of 7, which is tied up with skeletal muscle function and a brain function, and the fatty acid metabolism, and this is very interesting, and other various functions, insulin. There was a great paper on NAD and diabetes, as well as liver glucose metabolism. This is very interesting. This was so close to my heart. Proper NAD synthesis or upregulation enhances, not enhances, actually the opposite, decreases adipogenesis. And I kind of noticed it on myself. I noticed that since I started taking my NAD, it seems like my waistline trim, which is a great, great, great discovery. So as you see, not enough NAD, enhanced production of fat cells. Enough NAD, trim of the waistline. Again, not claiming or anything, this is from the paper, but this is my personal experience. And guys, that's about it. That's what I wanted to share with you. I'm going to stop sharing the presentation, and hopefully that was something new, and hopefully that was clear. If it is not clear, so please private message me on Facebook or send me messages if you want to. Other than that, thank you so very much for joining me for my first Facebook streaming and watch party, and have a great night. And thank you for everyone who allowed us on this Remembrance Day. I am a Russian, and a lot of Russians fell through the Second World War, millions, and my family members. So absolutely heroes and first responders. So thank you so much.